आवाज असेल नाही लाईव्हवर आवाज हा लाईव्हवर आवाज असेल ना
এখন মিলি ফেলে হয়ে যাবে
हाँ है यार ऑप्शन में लगी है और क्लिक कर अपना जाए
Good morning, everyone. I hope I am audible to all.
Yes, ma'am.
Yes, ma'am.
A very good morning to one and all present here. Pharmacists are the trusted source of knowledge, advice, and help. The way we cannot imagine patients without doctors, in the same way medicines cannot be imagined without pharmacists. A very happy World Pharmacist Day to all the distinguished pharmacists and to all the budding pharmacists, and of course to all of you. I am Ms. Arundhati Medhi, Assistant Professor in Pharmaceutical Chemistry at NEF College of Pharmacy, Guwahati. And I have the privilege for being your host today for this international webinar. Today, I request everyone to please mute. I request everyone to please mute their mics. I request everyone to please mute their mics. Otherwise, I will mute. Today, on the auspicious occasion of World Pharmacist Day, We, NEF College of Pharmacy, Guwahati, and NEF College of Pharmaceutical Education and Research, Nagaon, have come up jointly for the first time to organize an international webinar with the theme, Transforming Global Health. On behalf of both the institutions, I wholeheartedly welcome all of you. Before we get started, I would like to introduce all of you briefly about the NEF Trust and the groups of colleges run by it. So NEF, National Education Foundation, since its inception in the year 1993, has actively engaged in promotion of higher education in the entire Northeast India. NEF has been looking ahead for expansion, extension, and dissemination of education with more and more courses of modern studies. In its effort to encourage and inspire the young generation to opt courses of human and professional interest, the foundation has maintained constant touch with the modern trends in the field of education exploration, and thus it has maintained its pace and progress unabated in realizing its aim and objectives. It now stands as one of the premier organizations of the northeastern region of the country by virtue of its devotion and service to the society as a whole. The institutions, which are group of institutions which are run by the NEF Foundation Trust are at present are NEF Law College Guwahati, NEF College Guwahati, NEF College of Pharmacy Guwahati, NEF College of Pharmaceutical Education and Research Guwahati, Nagaon, then ICRA Academy High School Nagaon. Now, let me speak and brief about the importance of today's date, 25th September, as observed as the World Pharmacists Day every year. Every year, a distinct theme is used for the day, with this year the theme being Transforming Global Health. The purpose of this day is to encourage the activities and advocate the role of pharmacists in improving health in every corner of the world. This year, amid the corona pandemic, we are celebrating this event to express the solidarity to the important part of the healthcare team, the pharmacist. During this pandemic, apart from the doctors, nurses, and other healthcare workers, the pharmacists all over the world have worked day and night offering their selfless service towards the mankind. We express our deep gratitude and acknowledgement to each and every pharmacist all over the world. So in today's event, we will witness 3 outstanding lecture sessions from 3 dynamic and influential leaders of their respective field who are our important resource person for today's event. So I welcome all of you once again. Now, without any delay, I request our honorable and respected Dr. Zakir Hussain sir, Director, NEF Group of Colleges, to please officially inaugurate today's webinar with his majestic words. Sir?
Good morning. Am I audible?
Yes, sir, you are audible.
Namaskar. Respected guest of today's international webinar, Dr. Ashutosh Sharma, Director, Regional Department of Bioengineering, Monterrey Institute of Technology, Mexico. Dr. P. Ramalingam, Professor and Director, Ragavendra Institute of Pharmaceutical Education and Research, Andhra Pradesh. Dr. Amitava Ghosh, Unicorn Academy, Australia. Principal of NEF College of Pharmacy, Dr. Raja Chakraborty. Principal of NEF College of Pharmaceutical Education and Research, Nagaon, Dr. Devapratim Dasgupta. Distinguished participants, faculty members of different institutions, and my dear students. We all know World Pharmacists Day is celebrated on 25th September every year to promote awareness about the importance and role of a pharmacist. On this day, the main aim is to showcase the pharmacist and their positive effect on health. The theme of World Pharmacists Day 2020 is Transforming Global Health. Pharmacists transforming global health is the focus of World Pharmacists Day 2020. As frontline healthcare professional, pharmacists are facing an unprecedented workload during COVID-19 pandemic in pharmacies, hospitals, and other health establishments. I offer my sincere gratitude and thanks to all the courageous pharmacists on the occasion of World Pharmacists Day. I offer my deep sense of gratitude to Dr. Ashutosh Sharma, Dr. Ramalingam, and Dr. Amitava Ghosh for being with us in this virtual setting and hope their cooperation in future. I also offer my sincere thanks to the organizers for taking the pain of organizing this program today on this occasion. I sincerely believe that the deliberation will be delivered by the Dr. Ashutosh Sharma and Dr. P. Ramalingam, and as well as Dr. Amitava Ghosh will be helpful to our students community a lot. They will be enlightened with the new avenues and new innovations in the pharmaceutical industry. With this word, I conclude my lecture. Thank you. Thank you very much. Wish you all the best. Happy Pharmacist Day. Thank you.
Thank you so much, sir, for your kind words. Thank you, sir. Now, moving on. Now, I would like to request Dr. Pratim Dasgupta, sir, Principal, NEF College of Pharmaceutical Education and Research, Nagaon, to share his ideology on the occasion of World Pharmacists Day among our participants. Pratim, sir.
Thank you. Am I audible, Arundhati? Am I audible? Hello? Am I audible? Hello?
Yes, sir, you are audible.
Yeah.
Yes, sir, you are audible.
Yes, Dr.
Good morning. Good morning, respected Dr. Zakir Hussain, Chairman, NEF Trust and Director of NEF Group of Institution, Ms. Farhana Ahmed, Assistant Director, NEF Group of Institution, Mr. Nurul Haq, Dean Administrator. Sorry for the disturbance. Dr. Raja Chakraborty, NEF College of Pharmacy. I welcome the three distinguished speakers from different parts of the world. In this auspicious day, inconvenience, some network issues is there. Today in this auspicious day, I would like to mention the name of Dr. Mahadev Lal Shroff, the father of Indian pharmacy, whose contribution is really a big achievement. And due to his dedication, we can see us where we are today. Today, in this year, particularly this 2020 celebration of World Pharmacists Day is very significant in respect of the situation that we are facing today. The significant situation of this pandemic COVID, we can see the contribution of the pharmacists, and I really salute to their contributions. World Pharmacists Day is a day to be integrated throughout the world, all the pharmacists, and to take the pledge of to serve this community with our full dedication. In today's seminar, we are very much lucky to have the three speakers from the three different parts of the world, and I hope that their dedication and their objectives of this their session will be very much useful to all of us, and we will carry something from them in today's session. With this, I conclude today's my speech. Thank you all.
Thank you, sir.
Arundhati, you can continue. Yes.
Okay, thank you, sir. Thank you, sir, for your words. Next, I would request Dr. Raja Chakraborty, sir, Principal, NEF College of Pharmacy, Guwahati, to kindly share his ideas and thoughts on global health in context with today's webinar and inspire our young pharmacists with his valuable words. Sir. Yes. Yes, sir. Yes, sir, you are audible.
Thank you. First of all, I just want to say a wonderful good morning to all. Our beloved and our source of inspiration, Dr. Zakir Hussain, the Director, NEF Group of Colleges, Principal of NEF College of Pharmaceutical Education and Research, Nagaon, Dr. Devapratim Dasgupta. Our today's resource person, Dr. Ashutosh Sharma, Director, Regional Department of Bioengineering, Monterrey Institute of Technology, Mexico. Dr. P. Ramalingam, Professor and Director, R&D Division, Raipur, Andhra Pradesh. And Dr. Amitava Ghosh, CEO and Founder, Unicorn Academy, Australia. Our all the faculty members, dear participants, and organizing committee members. I wish all of you a happy Pharmacist Day 2020. We are celebrating this day with the theme Transforming Global Health. And definitely our speakers, excellent speakers, excellent resource persons will address all the participants, will enlighten them with their expertise and experience. I just want to offer this my gratitude to all the pharmacists all over the world who are helplessly working in this pandemic situation without carrying their own. And also all the pharmacists who are working dedicatedly to make this profession a respectful one and maintain the respect of this profession within this society. In this World Pharmacists Day, definitely we'll be celebrating in different ways, but this is the first time we are celebrating the World Pharmacists Day in a virtual basis. So we have a lot of plans for this in coming time, definitely. But in this regard, we are very much fortunate to have all the renowned speakers with us, and definitely they will enlighten us in this regard to all our participants. Without wasting much time, let's come to the actual session of our excellent speakers who will be coming up in time. Thank you very much to all.
Thank you so much. Yes, sir. Thank you so much, sir, for your valuable words. Now, remembering the lines of Mahatma Gandhiji, who says, "It is health that is real, and not the pieces of gold and silver." Now it's time to hear from our eminent resource person of today's international webinar. Before we dive deep into their words and enrich our knowledge bank, I would request Mr. Sorajyoti Deka sir, Assistant Professor, NEF College of Pharmacy, Guwahati, to please introduce Dr. Ashutosh Sharma, Director of Regional Department of Bioengineering, Monterrey Institute of Technology, Queretaro City Campus, Mexico. Deka sir, please.
Thank you, Arundhati ma'am. Thank you, Arundhati ma'am. So, good morning, everyone. On behalf of NEF College of Pharmacy and NEF College of Pharmacy, Nagaon, I welcome all of you for this international webinar on World Pharmacists Day. Along with that, I am also inviting all the dignitaries, all resource persons. So first of all, I have got the opportunity to introduce one of the most eminent personalities from the field of biotechnology, Dr. Ashutosh Sharma sir. So let me introduce him to all of our virtual participants who are present with us in this webinar today. Dr. Ashutosh Sharma had completed his BS and MS in biotechnology from the Guwahati University, Gwalior. Later, he did a PhD in Mexico on a bilateral project between Morelos State University, Mexico, and Arkansas State University, USA. After the completion of his PhD, he started a postdoctoral fellowship at the National Autonomous University of Mexico, UNAM, which is a world-ranking university coming under the top 100. And later, he moved to join Monterrey Institute of Technology, which is also a world-ranking university with a rank of 155 in 2012 as an assistant professor. Previous to his PhD, he has worked at several prestigious international research institutions in India, such as IMTech, NDRI, CCMB, and DWR. Within 4 years of trajectory, he was directly promoted to full professor category, being the youngest in the Queretaro campus. Since the beginning of his career, he holds academic leadership positions. During 2013 to 2017, he worked as Biotechnology Program Director and later was invited to hold the position of the Director of Regional Department of Bioengineering. Currently, Dr. Ashutosh Sharma sir led academy and research in the field of bioengineering, including biotechnology, food sciences, and agronomy bachelor and PG programs. He is a member of prestigious national researcher councils, currently as level 1, 2 of 4. He also participates as member reviewer of editorial boards of more than 20 international scientific JCR index journals. He has been invited in more than 37 national and international conferences as speaker and keynote speaker. His scientific contribution includes more than 75 original research articles, books, chapters, books, and patents. He is also the founder, director of New Center of Bioengineering at ITESM Queretaro campus, where he has established state-of-art facility, research center, and consolidated a highly productive scientific group. He has received various prestigious scholarships and recognitions throughout of his career. Dr. Ashutosh Sharma sir has participated in numerous academic activities and international scientific associations and have been consultant for a number of government and private sector scientific projects. So today he is with us as a resource person. So I welcome Dr. Ashutosh Sharma sir on this virtual dais and to share some of his experiences and knowledge on this graceful day of World Pharmacists Day. Over to you, Dr. Ashutosh Sharma sir.
Thank you very much, Dr. Deka. Very good morning to everyone. Would you allow me to share my presentation, please?
Yes, sir.
Can I be heard?
Yes, sir. You are audible. Just give us few minutes, sir.
Sure.
Sir, you can share your screen. Kindly click on the share option on your desk. Zoom.
Yes, it's still, I can't share. It's saying that host disabled participant is screen sharing.
Just a minute, sir.
Yeah.
Yes, sir. You are made co-host, sir.
This will stop the screen sharing.
Sir, you are. Yes, sir.
Yes.
Sir, you are given permission.
Now I can. Now I can. Thank you. I can. Here you go.
Yes, sir. It is visible. Sir, your. Yes, sir. Screen is coming full, sir.
Yes. Okay. Fantastic.
Okay, sir. Thank you.
Okay. So, well, again, thank you. Thank you very much, Dr. Deka, for your kind introduction. I'm very honored to be here. Well, again, good morning, good evening, good night as the case may be, because here in Mexico it's 12:30 a.m. And I hope that most of the people, they are connected from different part of the world, right? And I would like also thank to the organizing committee for organizing this pleasant event in this special occasion of Pharmacist Day. My gratitude to Dr. Zakir Hussain, Director of NAF Group of Institution, Mrs. Farhan Ahmed, Assistant Director of NAF Group of Institution. Also, I'm very thankful for Dr. Radha Chakravarti, Principal of NAF College of Pharmacy, and Dr. Devapuritam Dasgupta, Principal of NAF College of Pharmaceutical Education and Research. Thank you very much for having me here, and I hope you all are safe and healthy. Well, I'm going to discuss about the process of drug development from plants, and I'm very sure that everybody here have some interest in the pharmaceutical sciences. So medicinal plant lies in most of these area of pharmaceutical research. And basically this talk is about the recent panorama about the medicinal research focused on medicinal plants. And India, Mexico, China, and many other countries who has a very rich culture of medicinal plants. So we know very well the importance of this important natural resource. How do medicinal plants help us, first of all? Well, plants have been utilized as medicine for thousands of years. These medicine initially took the form of crude drugs such as tincture, teas, powders, and other herbal formulation. The specific plants to be used and the method of application for particular ailments were passed down through oral history. Eventually, information regarding medicinal plants was recorded in herbals. In more recent history, the use of plants as medicine has involved the isolation of active compounds, purifications, characterization, such as the case of morphine in the 19th century, from the opium was extracted. It has been estimated that there are approximately more than 400,000 medicinal plant species of angiosperm at least, but for most of these, only very limited information or knowledge is available. Three approaches which are closely related to diet, medicine practices, and scientific research like phytochemical analysis and so on can be adopted to explore the value of herbal preparations. And based on the experience from the random trials and observation in animals, ancient people acquired the knowledge of using herbs for treating illness. So basically that plants were our medicine. In this manner, Chinese herbal medicine, Indian herbal medicine, which were highly developed in ancient time in China, Japan, Korea, India, are still influencing the modern healthcare. The World Health Organization, WHO, estimates that herbal medicines provide primary healthcare for approximately 3.5 to 4 billion people around the world. Like 50% of the population get benefited from these herbal medicines. It is very interesting that drug discovery from medicinal plants has played an important role in the treatment of cancer. I'm just taking one of the example. There are so many illnesses, but cancer is very, very interesting and very important disease. Indeed, most new clinical application of plant secondary metabolites and their derivatives over the last half century have been applied towards combating cancers. Numerous types of bioactive compounds have been isolated, including alkaloids, coumarins, fatty acids, flavonoids, lignans, terpenoids, and so on. Several of these compounds are currently undergoing further investigation, and today approximately 80% of antimicrobial, cardiovascular, immunosuppressive, and anti-cancer drugs are of plant origin. Some of these examples you can see in the picture, very famous medicine, aspirin, morphine, and also the curcumin. There are so many medicines which have been extracted from the plants in direct or indirect way. Well, since 1990, there has been 22% increase in cancer incidence and mortality, with the four most frequent cancer being lung, breast, colorectal, and stomach. And there are four more most deadly cancer, being lung, stomach, liver, and colorectal, depending on the country and geographic locations. But this is like universal data. With the advent of high throughput screening assays direct towards these targets, known compounds from medicinal plants may have shown promising and possibly selective activity. And this is what we are interested in normally. And several known compounds have been isolated from traditionally used medicinal plants. 80% of the drugs which have been developed from the medicinal plants, 80% of these plants, they had a history of traditional use. So the drug discovery from medicinal plants has played a very important role in the treatment of cancer. And indeed, most new clinical application of plant secondary metabolites and their derivatives over the last half century have been applied towards combating cancer. The area of cancer drug discovery has been challenging, and medicinal plants has played a very important role, as we know that carcinogenesis is a multi-stage process by which a normal cell is transformed into a cancer cell. And this transformation implies a lot of different steps and long process, typically from DNA damaging, during which cell proliferates, proliferation is increased, progression, genetic alteration. So chemoprevention is another strategy which is new, and research is going on. WHO has already declared a list of compounds or herbal products which can be used as chemopreventive agent. So this is another new area. And some of the data which is quite interesting here, I'm putting India on the top, being one of the most diverse country, with more than 705,000 different plant species in traditional use, which is like half of the Indian native plant species. Then we have China. They have approximately 1,000 plant species are commonly used in Chinese medicine. And Africa, there are 5,000 plant species have been reported, but due to lack of research or the information, most of the information have not been explored. And Mexico is one of the fourth mega diverse country in the world. And in Mexico, they have more than 6,000 medicinal plants in traditional use, and out of which 400 plants have been commercialized and 100 of them are cultivated. This is just the data to understand, because my talk is about the necessity and current perspective of medicinal plant research. So anyway, another interesting data that WHO since 18th, for example, 80s, WHO did not recognize the value of medicinal plants until unless there were some advancement in the treatment of the diseases by different molecules which were extracted from plants. So finally, WHO, I think now they have 4 different monographs, if I'm not wrong, and which comprise more than 20,000 different medicinal plant species, which is considered to be promising to prevent and cure different kind of diseases. So how drugs are developed from medicinal plants, right? Because there are many ways that plants provide different drugs, and there are many myths, because many people think that medicinal plants are just using the parts of the plants. So there are phytomedicines, there are homeopathy, there are combinatorial chemistry coupled with the pharmaceutical. So there are so many things which can be done by plants, and this also create a lot of complexity in the structure to be able to develop medicines from plants. Initial success stories in new drug discovery came from medicinal chemistry inventions, which led to the need of development of higher number of chemical libraries through combinatorial chemistry. This approach, however, was proven to be less effective in terms of overall success rate. The second source of natural entities for sure are the compound from nature. Before the advent of high throughput screening in the post-genomic era, more than 80% of drug substances were purely natural product or were inspired by the molecules derived from natural resources. what we call them semi-synthetic analogs. An analysis into the source of new drugs from 1980 to very close to 2010 reveals that almost half of the drug approved since 1994 by WHO were based on natural products. And during the year of 2005 to 2007, 13 natural products related drugs were approved by FDA. And there are various examples of development of new drugs from the plant sources nowadays. In general, there are 6 classes of source of natural entities. The 4 classes are botanical. We can put them fungi, bacteria, marine sources. And in addition to these 4 classes, modern pharmaceutical chemistry added 2 other categories of man-made substances like synthetic chemistry and combinatory chemistry. Of these natural sources, botanical sources are the one we are discussing today. The botanical sources are known to provide the following classes of natural entities, which I already explained here. And unlike synthetic drugs, this is like a picture, the process how the drugs candidate are identified, then they go to clinical. The process looks very simple and few steps, but it can take decades. And that's the reality. So drugs derived from the herbal medicine are developed by isolation, identification, and then standardization, and then the trials, toxicological trials, pharmacological trials, pharmacokinetical trials, drug delivery trials, and finally the candidates will be selected, and they will go to the clinical trials depending on the extract, depending on the molecules, depending on the compositions. So the complexity is very high in all this process. Okay, whether we know the plant from many years, hundreds and thousands years of use, but the development process and final product to the market may take many decades, which is the case. For example, in 2005, one Chinese scientist received a Nobel Prize in chemistry, a pharmacist, Tu Juju, and she got this prize for the development for anti-malarial drug after 35 years. Okay. Anyways, so what we are seeing nowadays that a lot of people work with medicinal plants. A lot of people works. We have more than 60,000 plant species all around the world. 20,000 have been studied, but very few drugs have been developed. And why? This is the reason. This is the question, and the reason of this talk, that nowadays multidisciplinary approach should be employed strategically, and that's the only way by which we can develop drugs faster and quicker. One of the areas which is very promising in the field of medicinal plant research is the herbal genomics. New terms, we don't have more than 10 years with these technologies and sequencing capacity of the transcriptomes and the synthetic biology, microRNA analysis. Still there is a long way to go, but there are many advancements which is very important and promising. For example, in this manner, I'm putting like 6 different steps here, 6 different possibilities of herbal genomics. For example, with the developments of biotechnology and genomics, sequencing, whole genome sequence units or capacity technology, several species like Ganoderma and Salvia, they have been recently studies and whole genome have been published, and a lot of the biosynthetic pathway had been declared for the first time. Okay, the defining a molecular identity, for example, DNA barcoding is another very important tools. I will show you one real case where the DNA barcoding technique is very important to to identify the high quality and correct material to start your research. And in the part of molecular breeding, for example, the herb breedings is still a new area. Most of the breeding programs have been focused on the food crops, not on the medicinal plants. But yes, this is the area where we need a lot of efforts. Herbal, the gene bank, the herbal genetic information have been, being accumulated with the increased speed in last 15 to 20 years. And we have, nowadays at least few platforms where you can find out some genetic information, transcriptomic information, DNA barcoding database, where you can at least find some of the information. Still, this information is not structuralized. It does not have the proper structure. There are so many gaps, but still, I would say that it's a good advancement. Herbal synthetic biology, synthetic biology, the editing of genes and the genome, it's a very promising area. Some of the research have been done nowadays. The cannabis sativa have been studied, in different countries. They have been trying to modify these compounds, and some other plants, for example, have been modified genetically to produce valuable compounds and to understand the mechanism of action, because most of the metabolism biosynthetic pathways still are not clear. Another term is geoherbalism. It's another term when we are talking about a superior plant species in a particular geographical location, right? Understanding its environment and understanding the right conditions to grow this plant is also very important. Most of the research nowadays, we are focusing in the pharmaceutical phytochemistry area, most of the articles, most of the publications, but need to include, omics technology and genomic herbal technology and other technology I will also mention. For example, the omics I just mentioned, now we are living in a post-genomic era, right? And the screening of herbal ingredients should be accelerated due to the development of related disciplines. In this post-genomic era, the pace of drug discovery from herbal medicine is likely to be hastened and become more efficient than ever. In this post-genomic era, by performing a comparative study on gene expression profile before and after taking the herbal extract, that is also a very interesting area. And even some studies we have recently seen, very few articles have been published, but miRNA regulation before and after taking a herbal extract or a plant-based molecule, it's also a very promising area. Furthermore, genes responsible for regulating the pharmacodynamics and pharmacokinetics of the drugs can be better understood. Omics is a cluster of techniques. It's a collective technology which allow us to study different kinds of molecules. We can study carbohydrates, lipids, secondary metabolites, proteins, genes, and genomes. Okay, so but every approach is different. Their expertise is different. And working with all these area, it's really complex thing. Okay, in this particular sense, for example, that only few genomes have been published in plants. There are big projects about whole genome plant sequencing projects are being going on. One is in US, they call it 1,000 genome, and another one is going in China about 100 medicinal plant species, and there is another one which they call 10,000 genomes. I don't know how many plants they are going to sequence, but these projects are, at least they are going to create some platform, because we have too much to learn and too much to explore, because plants are very complex organisms. Well, thanks to these genomics technology, because this will obviously expand the herbal resources, what we have been discussing about. And while scientists are deciphering these genes in humans and the animals, they can also do same on plants of interest, right? If you compare the genomes which have been, investigated or developed in the field of microbes or microbiology, or with animals or insect or arthropods, you will see there's a lot of information available. But when it's come to medicinal plants, the very little information is available. In China, for example, I already mentioned there are some very promising projects are going on. In salvia, for example, the most of the genome have been the salvia miltioryza have been explored, published, and a lot of work is going on to understand biosynthetic pathway or different secondary metabolites of higher plants. Metabolomics, this is one of the very promising area which I personally do, and we have a laboratory where we work the metabolomics of endemic Mexican plants. And this area, it's not old. We have like 10 years, 13 years when the metabolomics was defined as a metabolomics, where we use the different analytical method, GCMS, HPLC, MS, and so other techniques and NMR. So collectively, these techniques can help us, to understand different kinds of molecules, characterize them, elucidate their structure, and finally having the right molecules or extract. Another very important part of this medicinal plant research is development of antiviral drugs, which nowadays we are having a pandemic, we are having a COVID-19, the situation of this pandemic. And we know that we don't have enough molecules, antiviral molecules. There have been a lot of research and effort in antibacterial, antifungi, antifungal molecules, but nowadays we are still fighting to have potential antiviral molecules. And this is not the case only just COVID-19. Viruses are difficult to treat, and vaccine seems to be the most common or traditional way to treat virus nowadays. But there must be some molecules which we can develop and treat different kinds of viral diseases. And there are so many diseases. We have still hepatitis B, we have still HIV, we have so many other viruses which may be the future or next pandemic. Okay, so there are so many viruses out there. But polio virus, for example, the vaccine took more than 55 years. HIV, we know since 35 years, still we are working on it. We know the genomes, we know the proteomes and everything, still we cannot treat it. So we need more effort, strategic efforts, where we can, work with plants which have potential to develop drugs against virus, right? Nowadays, for example, the vehicle, the mode of action, the delivery system is also very, very important, because the delivery changes the capacity of the molecules, absorption, and its effect. Okay, so nowadays there are a big category of products have been, developed, including nutraceutical, phytomedicines, and purified molecules, synthetic molecules. They all must be optimized, in terms of their concentration, to have their maximum benefits from its derivation, the concentration, which can reduce the toxicity, whatever it is, So, for example, nanotechnology is one of the potential technology which is allowing us to develop different kinds of delivery therapeutical targets in different tissues, and in trials also it's helping a lot. There are so many things which can be used. There could be polymers, biodegradable, natural or synthetic polymers, microcapsule, lysosome, lipoproteins, micelles. There are so many other options. Okay, this is one example of a Chinese medicine, for example, which have been tested for the influenza. And you can see that they are using more than 15 different kinds of plant species to treat one virus, and still these medicines are not that efficient, right? So there is no other, you already know in Ayurveda, the news we have been seeing, the research we have been going, we are again focusing on the plants which we know more than 500 years, and again we are depending on the immunomodulators and other kinds of plants which increase the immunity, but we don't have molecules which can kill the virus, right? So this is one of the most potential area where most of research should be focused on strategically and beyond the boundaries, obviously. Now I will talk about one real case where we have been facing a different kind of problems, because we have a very promising plant species. We have been working since many years with this plant species, but we have several, problems, and some of the problems which I will mention today. This is Galphimia glauca, basically endemic Mexican plants. It grows in all over the Mexico through Texas, Arizona, and up to Brazil. So basically, this plant is used to treat nervous system disorder problem, depression, anxiety, and for allergies and so on. And this plant have been first reported in the beginning of in 18th century, when the Mexican revolution was going on, and the soldiers used to take the extract of this plant in the post-war syndromes or problems. So it was used as an anxiety tranquilizer or natural tranquilizer. And since then, this plant have been used in homeopathy in Germany. And in Germany, you will find a lot of product in the field of homeopathy. And some of the products have been developed in UK and Europe. And the plant have been spread all over the world, in Japan, in China. It's very difficult to regulate the botanical material nowadays, but some regulation are going on. So this plant basically is from Mexico, and we have been working on it in try to understand the properties and pharmaceutical applications. In one study, we have collected this plant from 5 different states. 5 different states, and you can see the 1, 2, 3, 5. They are from different region of Mexico. Mexico is have if I'm not wrong, 29 different states, and these collections, were made. We identify these plants, we submitted in the suitable herbarium, we got the voucher, all that. And morphologically, this plant is very, very similar, extremely similar. And by morphology, it's very difficult to make any conclusion. Whatever we collected, we tag them as a Galphimia glauca. We got some confusion because we did some chemical analysis, for example, and we have some markers which I don't know why I'm seeing these lines over my presentation, but anyways. For example, this is the HPLC, and here you can see the 2 populations. One is from Guanajuato, another was from Queretaro, where I live. In these populations, you can see the 5 peak profile was the extract, methanolic extract, and these 5 peak profile is our marker. And these 5 peak profile represent the 9 triterpene molecules which we have published in 2004. These molecules we reported 2004, then further we reported in 2008 the work, and we had identify and elucidate the structure and all that. And we knew that this compound is only represented in these 2 populations, and these other 5 populations, they did not have any of these peaks. And the TLC, the TLC, the thin layer chromatography method, by which we could, we have a standardized method. We have this again, the 3 bend profile in the case of triterpenes, and this is a purified compound. And you can see that most of the plates here, basically only the QJ and the GM are the one which has the Galphimines profile. Galphimines are the triterpenes which we have been reported. And in the other population, you won't find this. There are some same polarity compound, but finally we try to confirm this by NMR, where we have purified compounds from the protons, which are the protons from basically vinylic protons in the purified compounds. And the Galphimines, they are A, B, C, D, E, I, F, 9 different compounds. So here we can see that only the purified compounds and the population which I already explained, GM and QJ, were the one who had the signals which represent the triterpenes. And finally, by in vivo models in rats, the elevated cross model, we tested our hypothesis that if if the compound is not here, then we have to validate this properties in rats. And finally, we find out that yes, indeed, the populations, the QJ and the GM were the only active populations. And here you can see the diazepam were used as the control. Other populations were not active. In this case, for example, the sedative test, when you calculate the time of rat, sleeping, again, the activity of these GM and QA were the significant, other activities were not. So we made the conclusion by the in vivo and in vivo models that definitely the compounds we have reported, for sure they are present only in 2 populations. So why everybody in herbarium, for example, tagging this plant species as Galphimia glauca? So there must be some chemotypes, there must be some geographically inducer or elicitors, there must be some reason that only 2 populations producing these compounds, which we, by multivariate analysis, we tag them as active populations, we identify the signals, and finally so on. Now we are working on the methodology which UPLC nowadays, and trying to identify new molecules. But finally, when we had this problem, we tried to do DNA barcoding to basically find out if there is any genetic variations in this plant species. And finally, surprisingly, we could see that yes, all 7 populations they group in 3 different clades, and the populations which have already sequenced by a Chinese group in PubMed, it does not represent sequence from our analysis. And obviously the population of QJ and GM genetically they are similar than these CT and MC, MT, MM group clad. Okay, so genetically we could identify there is a difference. Another marker which we use, RPC1, also tells us the same story. So finally, we identified that this plant is not a single species. In this genus, we have more than 26 different species. And nowadays, we resolve this mystery because so many products have been developed from this plant in China, in UK, in Japan, in Germany, and many people have collected the wrong material. So what they are selling, what they are developing, I don't know what kind of properties they are offering to the clients, but at least we know by the help of the marker, by the help of metabolomics, that the real population, it is in this particular region, and these are the species, and it comprises at least 26 different species in this particular Galphimia genus. So the moral of the story that we have to work in many aspects. If we want to develop medicines, then we have to work in different aspects of the plants. We have to do most of the things which we can perform. I know for a single laboratory or single group, it's not possible, but at least we should do in collaboration or whatever is possible for everyone. But we have started a multidisciplinary group of medicinal plants where we are trying to employ most of the techniques which we can offer and afford in terms of all the genomic analysis, miRNA analysis, and molecular docking. I have been recently collaborating with some of my colleagues in India. Micropropagation is another area, larger scale field production is another thing. Phytochemistry, obviously, the most traditional, most widely used, molecular biology and metabolomics. So we are trying to work in all this aspect with this particular plant species. And just imagine, we need how many years and decades to investigate this plant to be able to develop one drug or compound from this. Okay. I don't know how much time I is left. Anyways, just quickly going to pass from this. For example, these are the phytomedicine which have been registered in last 15 years in Mexico. And you can see that these medicinal drugs, these phytomedicines, most of these drugs, they are from export-import business. Basically, they are from some national companies and international companies. You already know that. And out of these 223 different phytomedicines registered and commercialized in Mexico, you can see they are just belonging to 45 most commercialized plant species. And we don't have any one single endemic Mexican plants here. So basically most of the phytomedicine research or phytomedicine product, they comes from Europe, and in Mexico they commercialized by the license and by the patents and other things. But there's a very lack of interest in this field. This is the plant, for example, the same plant I present to you. It grows in a very desertic conditions. You can see that the conditions of the soil, the stones. And so we did another project just not to lose the control, because the plant is, extincting from, it's going to be in the endangered list by the national authority very soon, because due to urbanization and development, and we are losing its geographically located. It's very difficult to find this plant nowadays. So then we had to work with the micropropagation and other commercial, traditional sexual reproduction methods. I'm not agronomist, but we had to do it because otherwise we were about to lose the plant. So finally, we achieved this method. Now we are able to, propagate plants, because we are working with the large scale standardization of phytomedicine extractions, phytomedicine extract. So we need this plant in a very high amount, and that was not possible by traditional collections. So basically nowadays we have enough material. We are improving the method by micropropagation. Then we try to develop a project, an integral project with rural development and with the the academia. So we involved the senator of the another state. We got the John Deere, the Monsanto, the Pioneer Seed Company directors of Mexico. We make a team together, and we rented a place, and we involved the farmers. They used to cultivate different plants, but not medicinal plants. So we put the Gulfimia glauca in the field by using drip irrigation and the tunnels and all that agriculture applications to have wonderful, beautiful, healthy plants. And finally the project was very successful. It was a pilot study, and nowadays we are able to save this plant, conserve this plant, but we are also having a plant of very high quality. Nowadays we are working on how to induce the production of these triterpenes. This is one of the projects we are working with, using different technology, right? Light and other kind of elicitors. You can see that this project, we grow Gulfimia with many other horticulture plant. For example, you can see lettuce and papaya and cucumber, maize, sunflower, chilies. Many different kinds of vegetables and fruits were developed together with the medicinal plants. So the project was full success. We got all the support from the private companies. We did not invest not a single dollar here. All were supported by the community, by the people, by the companies, and by the university. Several papers we have published in different areas, right? But we are still working on different aspects of this particular plant species, to counter the problems by multidisciplinary approaches. So finally, well, we have different challenges nowadays. Many of them I have already mentioned. In this era of rapidly advancing science and technology, there is a tendency to ignore traditional value and knowledge, as well as traditional medicine as large. Although in this post-genomic era, we have great opportunities for screening active compounds from medicinal plants, and we should be aware of traditional knowledge in an attempt to discover drugs derived from herbal medicines, not just depending on the traditional use and traditional history and traditional beliefs. We have to prove it by scientific evidence, and many medicinal properties of plant species were revealed from experience accumulated from a long history of use in many traditional herbal therapies and cultures. And generally, the success rate of synthetic route for developing new medicinal agent, it could be even 1 in 1,000 trials. And however, the success rate with search for new therapeutic moieties based on medicinal plant used in traditional medicine system can be increased as high as like 1/4 or more. We have to improve the techniques, technology automatization, extraction methods. We have to improve all that. Another one of the biggest problem is the active compound isolation, the quantity. One of the reason of doing micropropagation is to have enough material, because we did not have material. And this plant is very difficult to localize. It's very difficult to localize, and very rare location you will find this plant, the plant which produce the compounds. You will find so many other varieties and species which are available in other states, but this particular population is the species is very difficult to find. Anyways, I think I already passed my time. So, well, thank you very much for the invitation. I'm ready for your questions. This is my email and my Facebook and Twitter, and all yours, sir. Thank you very much for your attention, and I'm ready for a question if you have any.
Yes, sir. Thank you very much, sir, for your insightful knowledge, for very informative information on medicinal plants, then medicinal plant discovery, then herbal genomics. So it is a huge knowledge that we have gained today. We thank you very much, sir. It is a privilege to have got to listen from you today. Thank you so much, sir. Now,
thank you very much.
Now I request our participants to please ask questions or any doubts through their chat box. You can write your questions in the chat box and you can ask, sir. Participants, please write your questions, your doubts in the chat box.
It seems there is no question. It's a Friday morning. People are waiting for the weekend, I guess.
Yes, sir. Yes, sir. We have one question, sir. How we attribute nanotechnology to medical metabolites, sir?
Okay, nowadays, well, there are several ways, but nowadays there is one important focus is about delivery system, how we can attach the molecule, the extract, the purified molecules to some delivery vehicles. So this is one of the most promising area. We have already working, we have published the nano particles synthesis and the characterization and the in vitro test of different kind of terpenes to attach to be able to attach on some different kinds of nanoparticles. So basically, nanotechnology is the synthesis and characterization of nanoparticles. And here, being people interested in medicinal plants, we try to find out new vehicles, because in some plants or in some extract you can increase the capacity, property, or you can inhibit the toxicity. So there are so many advantages of using nanoparticles. Depending on the plant species, some plant species will respond, some will not respond. Basically, in our case, in case of Galphemia, for example, we have tested by copper-based nanoparticles, for example, and we had some good properties, enhanced properties. So most of the research is going on in this way, using nanotechnology with medicinal plants.
Thank you, sir. Any more questions, sir? Sir, can you please help me to, Deka sir, please help me to see the chat box.
Thank you, Arundhati ma'am. Any participants, if you are having any type of question, you can please raise your question.
Oh, please go on. We still have time.
Yes. Yes, sir. all participants, rarely we get the opportunity to meet with resourceful person. So kindly utilize your opportunity. Requesting
It's
to all
1 in the
participants.
morning, Mexico, 1 a.m.
Yes, sir. Here it's 11:40 a.m. morning in India. Sir, we have one question. The question is, how flavonoids as secondary metabolites has more importance?
How flavonoid as secondary metabolite has more importance? I will not say more importance because there are many categories of secondary metabolites. We have alkaloids, we have terpenoids, and we have flavonoids. So basically, they have different properties. I won't say that one is important than other one. Basically, for example, in my case, basically I'm more interested in terpenes molecule, terpenoids. But yes, in some cases, for example, flavonoids are known for antioxidant capacity. In some plant species, many compounds, antimicrobial compounds, antiviral compounds, they are flavonoids. So it's very difficult to compare in terms of their importance. I would say all are important, but we need to know more molecules. Because if I'm not sure, we have identified close to 150,000 flavonoids molecules so far. And this list of molecules, if you compare the number of species we have on the land plant, a number of species we have of the gymnosperm, a number of species what we have in ocean or yes, ocean flora, its number is so high. So we need more efforts, more technology, and more research to identify more flavonoids, more alkaloids, and more terpenoids. That would be, I think, the more reasonable.
Yes, sir. So we have one more question. What do you think about the future scope in medicinal plant sector in 10 years in India?
Sorry, madam?
So the question is, what do you think about the future scope in medicinal plant sectors in 10 years in India?
Okay, very interesting question. Well, in India or all over the world, I think the situation is very, very similar. Being India, for example, India has one of the biggest pharmaceutical industry in the world. But again, we have to do more research. We need more research. We cannot just rely on one plant which we know since 600 years and doing research and research and research and research by this way, another way, this dimension, other dimension. And still we don't have any complete or any conclusion, right? Curcumin, for example, or turmeric, turmeric failed most of the clinical trials in terms of when they were about to release the product. It failed. Why? So we have to find out, the logic of doing research with the focus of product development. I find lack of strategy and lack of scientific profoundness when we are working with medicinal plant. We believe still so much in the traditional use, and all the importance were given that this was used in hundreds of years and this treat everything. And many things are going like that way. A big market have been created, a big market have been created of herbal products everywhere. I remember in Mexico 10 years back in Walmart, I don't used to see herbal products. Nowadays, there is a full section of hundreds of teas, hundreds of natural extract, hundreds of company. So there is a boom. There are companies which are taking the advantage of this tendency of everyone who want to use the natural products. But being natural doesn't mean that it is not going to harm you, or it does not mean that it's going to cure you. Without science, without scientific profoundness, without scientific evidence, we cannot prove that this is going to work. And you already know this is happening. Some people claim that I got the drug, I got the medicine, I got the capsule, and in clinical trials, they all fails. They don't have enough evidence. So we have a lot of marketing, lot of selling things, but we have little research. So we need more accurate research. We need multidisciplinary approaches like metabolomics, like genomics, like synthetic biology, all together to be able to develop products, right? Products which is really benefitable and also provide us the right information about the complex biological information of plants. Plants cannot move, they cannot speak, right? They look very simple, decent guys, right? But believe me, they are the most complex organism of this planet. Every species is unique, every plant is unique. So to understand them, we need to use new tools and technology. And obviously we have so many tools nowadays. So in 10 days, what I will see that we have to focus, hopefully the government, or there will be some strategy that people should focus in new technology, in the tendency, not just selling the product, just using the benefit of the traditional knowledge, because we know there are traditional knowledge, there are traditional histories behind many plants, but these plants are clinically failed or they don't have any scientific evidence of their importance. So when there is no scientific evidence, I think we should not promote those plants, right? So in 10 years, in every place, maybe in Europe, India, or Mexico, I think these multidisciplinary approaches must be employed in a very strategic manner.
Thank you, sir. Sir, we have two more questions from our colleagues. Future scope in herbal plants in northeastern region of our country.
Future scope in herbal plants.
Yes, sir. In northeastern region
Future
of
scope.
Yes, sir.
Well, it's a very open question. Future scope in herbal plants. Scope. This very famous word in India. Scope of this, scope of that. Everything there is evaluated in terms of scope. I think scope is everywhere and in everything. Basically, you have to find out the problem. What is the problem? You try to find out the right problem, and then you will have a solution which is going to create a scope. So basically in northeastern region, for example, I'd see the climate challenges or climate favored conditions. So being there, I never been in northeastern region, but I know geographically that there must be some challenging areas, but there will be some benefit of these regions. So I think being youngster, being pharmacist, being biotechnologist, you guys, try to look into it and find out some problems and let's, try to find out some solution, entrepreneurship. I always, try to convince to all the youngsters that don't look for the scope, try to create a scope, right? So scope is everywhere, everybody gets sick, right? Population is increasing. So if you ask me about scope about health, I think this is the brightest scope, right? You cannot say there's nobody going to be sick after 10 years. There will be more population, there will be more challenges, there will be more diseases. So obviously scope is there, but we have to grab right problem to create right scope.
Yes, sir. Thank you, sir. Our next question is, she's asking about sir, metabolomics research, something about metabolomic research.
Metabolomics is a good tool. It's a technology of collective techniques, right? Where you can, helpful by different analytical techniques like HPLC, gas chromatography coupled with mass spectroscopy, or liquid chromatography coupled with mass spectroscopy, nuclear magnetic resonance. Even now, you can also include the supercritical extraction methods. So there are so many techniques out there with the use of multivariate analysis, a statistical tool, and that they help you basically to try to identify new compounds, by try to separate or purify new compounds or old compounds. So basically, these techniques, collective techniques in metabolomics, it was referred in 2008 as metabolomics, not in. I completed my PhD in 2012, and when I started working in metabolomics, I remember in 2007 or 2008, if not remember, it was like published for the first time in one article that metabolomics are a new emerging tool, and they just collectively started calling metabolomics. So metabolomics is a technology within the omics group where you have lipidomics, proteomics, genomics, transcriptomics, and every omics basically is to study one particular molecule. In metabolomics, you study metabolites, and metabolites basically it could be anything. And most of the time we consider this metabolomic as bioactive compounds, natural products, or secondary metabolite. So I don't think, I'm not sure if I answered your question, but if you have another question, you can ask.
Thank you, sir, for clearing their doubts. Thank you so much, sir. So we have one more question. I don't know it is relevant or not. So how long does agrobacterium will present in plant after infection?
I don't know where this question come from, but I don't know basically about pathology. So basically, I think I'm not the right person to answer this question. How long it will get, present in the plants? What I know that being a bacteria, it could be present until unless the plant cannot kill it or defend it, right? But the question, I don't know if it is about the real natural conditions, it is a lab or tissue culture. So maybe I'm not the right one to answer this question.
Thank you, sir. Thank you so much for giving your valuable time. Thank you for this informative session and giving us so much of knowledge about medicinal chemistry as a whole. Thank you so much, sir. Now,
thank you very much.
Thank you, sir. Now, I would like request Mr. Sajidul Ansari Haq, Assistant Professor, NEF College of Pharmacy, Guwahati, to kindly deliver his vote of thanks to Dr. Ashutosh Sharma, sir. Sajidul, sir.
Okay. Thank you, Arundhati ma'am. Am I audible, ma'am?
Yes, sir, you are audible.
Okay. So first of all, a very good morning to all, and I wish you all a very happy World Pharmacist Day. On behalf of NEF College of Pharmacy, Guwahati, and NEF College of Pharmaceutical Education and Research, Nagaon, I feel honored and privileged to propose a vote of thanks to our honorable speaker, Dr. Ashutosh Sharma, sir. I would like to extend a very hearty vote of thanks to Dr. Ashutosh Sharma, sir, for blessing us with your presence. I would like to thank you for your valuable speech and sharing and enlightening us with your knowledge, expertise, and finding. Your talk on medicinal plants research, past, present, and future was very informative and knowledgeable. The way you have explained how a drug candidate identified from natural products and also about
Sajidul sir, you are kindly unmute yourself. Sajidul sir, kindly unmute yourself. You are muted by.
over to you, ma'am.
Yes, sir. Audible. Continue.
Thank you so much, sir. Thank you so much for your valuable time and for being with us. Thank you so much, sir.
We break for lunch.
With this, we will have a break of 1.5 hours. And I request all the participants to please be connected with us and join us again by 1:30 p.m. So Dr. Ashutosh Sharma sir has shared his email, then his Facebook and Twitter account to all of you, to all the participants. So whatever queries and doubts you have, you can note down this information from him, and you can ask your questions and you can connect to him through this. Thank you so much, sir. Okay, now we are having a break of 1.5 hours, and participants, I request you to please be connected to us by 1:20 sharply, and we'll start the program by 1:30 p.m. Thank you.